Great. All right. Well, hello, good morning. Wherever you may be joining us from, on behalf of the conference planning team, the NTSAD staff and board, we would like to welcome you to Research Day for the 2020 NTSAD Family Conference. Whether you've joined us in years past or this is your first time participating in the conference, and I know there's quite a few of you that is the case, we are so very glad to have you here with us. We would also like to thank our conference sponsors who are helping to make this event possible. And if we could show the conference sponsor slide, please. Great, so thanks to all of these organizations and foundations who have helped with this year's virtual conference. My name is Jamie Ring, and I became officially involved with NTSAD last July as a board member. But I've unofficially been engaged with the organization since 2005. I've attended many Imagine and Believe events over the years and the conference a handful of times as well. I've spent my 20-year career working as a patient advocate within the biotechnology industry, supporting rare disease treatment development. I'm a firm believer in the need to incorporate the patient perspective into treatment development, and I've seen firsthand the positive effects of collaborations between advocacy groups and companies in accelerating these treatments. I spent 10 years of my career at Genzyme, and I've spent the last 5 years at Spark Therapeutics, where I was involved in numerous activities related to patient advocacy for Luxturna, which was the first FDA-approved gene therapy for a genetic disease. Throughout my 2 decades of patient advocacy work, I have learned many things about the drug development process and what it takes to bring new therapies to those who need them. And I firmly believe that an educated and empowered patient community is paramount in the process, which is why I wanted to get involved and contribute to this exciting but complex time in NTSAD's history. This is my first time participating as a board member in a patient advocacy group. And honestly, it's the privilege of a lifetime to be able to be a part of an organization that has been so supportive for families, so steadfast in their mission, and as focused as NTSAD has been. Over the next hour and a half, you're going to have a chance to learn more about the drug development process from a clinical research perspective and a regulatory perspective. And panels like the one we're about to have and are going to experience are so useful in helping us all understand how research goes from bench to bedside. We're very lucky to have such esteemed guests joining us to help educate and empower us and help give us an overview to assist in building some foundational knowledge for the research process. So just a little bit about our panelists, but they're going to introduce themselves as well. Dr. Jerry Cox is an NTSAD board member and a clinician who has worked in rare diseases throughout his career, and he'll be reviewing the clinical research process with you today. And both Dr. Witten and Dr. Simpoku are volunteering their time as ambassadors from the FDA, an organization that is so very critical in the review, approval, and monitoring of new therapeutics. Dr. Witten is the Senior Medical Officer in the Division of Clinical Evaluation and Pharmacology and Toxicology at the FDA's Center for Biologics Evaluation and Research. And Dr. Simpoku is the Acting Deputy Director in the Division of Rare Diseases and Medical Genetics within the Office of New Drugs at FDA Center for Drug Evaluation and Research. I want just to remind everyone also that in less than 2 hours at 2 p.m. Eastern Time, there are going to be 4 disease-specific breakouts where you'll have a chance to learn more about specific programs in your disease area of interest and ask questions specifically about those research programs and clinical trials. And then I also want to make sure that you're all aware that Dr. Fran Platt, who's the NTSAD Scientific Advisory Chair, has provided a very useful general overview of the diseases themselves via a pre-recorded webinar. So please avail yourself of that important information at your convenience. That was sent out in an email from the team earlier this week. And thank you, Dr. Platt, for your ongoing support. On behalf of Sue and the NTSAD team, I'm going to spend just a few minutes providing a very high-level overview of the research landscape for NTSAD. Okay. So as you all know, NTSAD is one of the oldest advocacy groups in the country. And over the course of their 6 decades, they have been instrumental in funding and supporting foundational research for the NTSAD family of diseases. Throughout their history, NTSAD has funded 66 projects exceeding $4 million, which has since been leveraged to over $30 million in NIH funding. This is just a snapshot of the work that's been contributed to date. These are the active grants as of May 2020. And as many of you may know, in 2019, NTSAD underwent a comprehensive strategic planning exercise. And so we wanted to help educate you all about how the organization is poised to continue to build capacity and contributions for research in the future. And, I just want to mention that, when we think about the contributions to date from NTSAD, undoubtedly, these efforts have helped create and foster the current research landscape that we're going to learn more about today. So NTSAD, as you all know, serves, has a primary purpose of connecting families, researchers, clinicians, industry, and government, all in the spirit of advancing research and developing treatments. And so out of the new 2019-2020 strategic plan, a number of priorities were identified to help support these efforts. And so 3 that are relevant for our discussion that will be upcoming are: 1, to identify research priorities. So where should NTSAD be investing? What are the priorities that will help drive us to future therapies? 2nd, to define and advance NTSAD's role in enabling clinical development. 3rd, to advance earlier detection through advocacy and research, which is something that NTSAD has been doing throughout their existence. And importantly, what I wanted to note here is that there are currently task forces in place to define gaps and to develop plans at a tactical level to support these initiatives. And those task forces, in some cases, exist of some of the leaders in the field, which I think bodes well for how NTSAD will continue to support these research efforts moving forward. And lastly, NTSAD has a common goal to promote information sharing and collaboration amongst all stakeholders to help get us to our mission. And then I wanted to also share a view at the current state of research and trials. This is really incredible to look at this slide. This is not what the case was, as you all know, just a handful of years ago. And it really is with, without the decades of work that NTSAD has done in conjunction with all of you, the families that are making this possible. We're entering a new era of hope for sure. And that's why educating you all through today's efforts and ongoing efforts about where we go from here as we look at all the different programs that are being developed currently is really why we want to have this panel today and the research sessions this afternoon. And then lastly, before we begin the presentations, I know we're all still kind of getting used to this whole Zoom format. So just a few logistical reminders for you. As you probably know, you're all technically muted, so there's no way for you to speak up and ask questions in an audio capacity. But if you just hover down at the bottom of your screen, there should be a box that says Q&A, and that's where you'll be able to ask your questions. You can ask questions at any time throughout the presentations, and we'll work at the end. We'll have a Q&A moderated session. We will do our very best to get through as many questions as we can. I want to just remind all of our attendees that the NTSAD families really are our primary audience, so those questions will take priority. And if we don't get to your question for some reason, or if it's maybe a more personal question related to your specific medical case, please feel free to connect with the NTSAD team following this session, and we'll do our best to get you the information that you're looking for. Okay, so at this point, I am going to turn it over to Dr. Jerry Cox, and I will see you all in a little bit for the Q&A.
Great. Thank you, Jamie, and welcome everyone to this session. I'm particularly pleased to have the opportunity to speak with you today. As Jamie mentioned, I joined the board last year, and I've had a long career in both drug development and clinical genetics. I've been affiliated with NTSAD for several years, particularly the late onset Tay-Sachs and Sandhoff community. And I'm really happy to be able to share some of the insights and knowledge I've gained over the past couple of decades working in rare disease drug development. And as I thought about the focus of my talk, I thought really there are a lot of questions that families have about clinical trials. And so I've structured my talk around answering those questions. The first several questions really deal with information about clinical trials, what they're all about, what purpose they serve. The next few questions have to do with participating in a clinical trial. And the last few questions have to do with topics like expanded access, the impact of COVID-19 on clinical trials, and then probably the question we get asked all the time is, how long does it take to get a drug approved? So if you can stay with me through my presentation, hopefully I'll be able to answer that question at the end. So what is a clinical trial? A clinical trial is a study. It's an experiment really involving a drug or a device and humans. It could be healthy volunteers or it could be patients. And the study is designed to address specific questions about a drug. Being investigational drugs that have no widespread experience, we have to identify what is the right dose of the drug and how often should it be taken. Is the drug safe? What are the side effects? Does the drug work? What can we expect it to do if a patient starts taking it? Then finally, who should or should not be taking the drug? As we all know, there are some drugs that could be harmful to fetuses that should not be taken during pregnancy. There are also situations where people may be taking another drug that might interfere with the experimental drug. And there could be situations where people have liver disease or kidney disease that might interfere with the levels of the drug. So these are all part of the clinical development process to ensure that when a drug does get approved and is used widely, that the drug is safe and effective. Now, we don't do everything related to these questions in one trial. We do them in a staged manner that we refer to phases. And early phase or early stage trials are usually called phase 1 or 2, and then the later stage trials are phase 3 or phase 4. And the early stage clinical trials are really focused on getting the dose right and making sure that whatever dose we wind up using in patients is going to be safe. And in the early stage trials, we want to make sure that the drug we're using has biological activity. And the way we do that is by making objective measurements related to what the drug is supposed to be doing. And that would be related to changes in what we call biomarkers. Now, for the NTSAD-related diseases, this could involve levels of NAA for Canavan disease or levels of GM1 or GM2. for the gangliosidosis. But for many of these drugs, if they work on the underlying cause of the disease, we would expect that certain biomarkers would change over time. And then finally, the late stage trials are really to confirm what we suspect, namely that a drug is efficacious and that it's safe. And there are specific guidelines about how those studies should be carried out. So what does a clinical trial involve? Well, it starts with a clinical trial protocol, which if you're a cook, this would be like a recipe for a dish. If you work at a business, this would be like a standard operating procedure. It lays out exactly all the specific information about the clinical trial that should be followed so that a specific conclusion can be made at the end of the trial. From a patient point of view, patients should be informed that this is an experimental treatment and that there may be risks in addition to potential benefits, and that they sign a statement called informed consent if they're an adult, or an assent if they're a minor, stating that they understand that this is experimental and that there are risks. Similarly, the investigator for the study, who is typically a physician expert at a hospital, they also receive information called the investigator brochure that outlines all the information that's known about the drug, from its manufacturing to mechanism of action to results of animal studies, and then any clinical data that's been accumulated, so that they can decide whether they want to be the investigator for this study. To participate in the study, patients have to meet certain criteria for eligibility. So again, every study has a purpose, and some patients may have specific characteristics that make them more amenable to one study compared to another. There's also expectations that are laid out for study participation from the patients, namely that they agree that they would take the drug on schedule, because that's the purpose of the study, is to investigate the drug, and that they would show up at study visits where physical exams would be performed. There may be laboratory tests or other assessments performed over the course of a study, so that at the end of the study, the results can be analyzed and there can be an evaluation of the safety of the drug, as well as any kind of changes over time that occur with respect to potential benefits. With rare diseases, there's often no other drugs that are available for treatment. And so typically, once patients finish the first study, they may enter into what we call a long-term extension study, or once the drug is approved, they may enter a disease registry to continue to collect information about the effects of the drug. Now, there are several key people or institutions involved with a clinical trial. It begins with a drug company that has the idea for producing a drug to treat a particular disease, and they have to figure out a way to manufacture the drug, usually test it in animals, and design a first-in-human clinical trial, all of which is submitted to the FDA in the form of an investigational new drug application, or IND. The FDA reviews that information. If they consider it reasonable to proceed into human trials, then they give the approval, which is then sent to the hospital that the study will participate at. And the hospital will have a committee called the Institutional Review Board, or IRB, that'll review the protocol, and in particular the informed consent, to make sure patients know what they're getting into, that all the risks have been outlined. And then they will give their seal of approval to the study investigator, who will then start recruiting patients for participation in the clinical trial. So the clinical protocol, what does that involve? Well, every protocol has a series of objectives or purpose, as we mentioned. It may be for dosing or safety or efficacy or some combination of those. It'll have a trial design that'll explain how patients will be enrolled into the study, what doses they may be getting, whether there may be an additional group of patients that might receive a different treatment or standard of care, or potentially a placebo. And in the case of other treatments, they may be disguised in a way that no one in the trial knows which drug the patient is getting, either experimental treatment or some other treatment or placebo. And this is really important for particularly later stage studies where we want to eliminate any potential bias or what we call placebo effect, where patients may feel like they're getting better because they're getting treated, but in fact, there may just not be any active treatment that is making them feel better. That's what we call placebo effect. We need to distinguish that from the actual experimental treatment. With every study, we need to define how many people will be participating, which clinical sites, which countries. In the US, the regulatory body overseeing clinical trials is the FDA. In other countries like Europe, there's the EMA. In Japan, there's the PMVA. And if we have clinical sites in other countries, those respective regulatory bodies also have to review the study and allow the trials to continue. Every study will have a defined duration that will relate back to the study objective. So if you're just looking at dosing, it may be a relatively short trial. You're looking at clinical benefit, that may be much longer, on the order of 1 or 2 years. For every trial, there'll be certain entry criteria that patients have to meet, and there'll be a clear description of what type of safety monitoring will take place, what the endpoints are that will determine whether or not the drug is working. There'll be a list of study assessments that patients will need to complete and a schedule of visits that they'll need to make to the hospital. And then with every protocol, there's a statistical analysis that will be used to support the overall design of the study to make sure that there are enough patients of the proper characteristics and assessments that are enrolled in order to make a conclusive statement about the outcome of the trial. Now, as I mentioned, different trials have different categorical names. The early stage trials are considered phase 1 or phase 1/2. With small molecule drugs that you may take by mouth, those are often administered to healthy volunteers to get dosing information. However, with biological drugs that may have long-term effects or other effects that healthy volunteers may not want to be exposed to, that those particular drugs would start in patients. We usually look at a series of different doses where we enroll patients at lower doses first, and then if those are safe, we continue increasing the dose over time in different patient groups. We'll evaluate the safety, the dose level, and the frequency of dosing. We may use the drug levels in the blood, what we call pharmacokinetics, to determine what the appropriate dosing regimen is. Or we may also look at what we call pharmacodynamics, or PD. What is the effect that the drug is having on the body? So we may look at some dose levels that partially lower NAA or GM1 or GM2 levels, whereas other doses do a better job. And so combined with the PK and PD data, we get a sense of what the most appropriate dose and regimen is. Now, sometimes we can see some signs of clinical benefit within the first few weeks or first few months of treatment. And that's really a bonus. And that gives us some hope that we're on the right track, but it's really not the primary objective of the phase 1 or phase 2 study. The phase 2 or phase 2/3 study is the study where we want to demonstrate safety and efficacy usually at 1 dose. And this is going to be the study that's used to support the approval of the drug. And with these types of studies, they need to meet certain criteria that you'll hear about later in the session. They need to be adequate and well controlled. Adequate means that they need to be carried out in a large enough number of patients that you can determine the treatment effect. They also need to be conducted in a rigorous manner according to certain standards for clinical trials. They also need to be well controlled. There needs to be a reference group that we compare the effects of the drug to. And this is often a placebo control, but it may be standard of care. And in cases where the natural history of a disease is very well defined, it may be a historical control group. And regardless of which type of control group, there needs to be a clear what we call substantial evidence of clinical benefit. So a result needs to be statistically significant. It needs to be clinically meaningful to the patient. And the gold standard for these types of trials would be a double-blind, placebo-controlled trial where neither the patient nor the investigator knows who is getting drug and who's getting placebo, and that the main measure of efficacy is a measure of clinical benefit to the patient. And then finally, once a drug gets approved, there may be other patient populations, for example, different younger patients or different age groups or patients with special medical problems that need to be studied as well.
Hey,
So
Jerry.
yes.
It's Jamie. I'm just giving you a 5-minute warning.
Okay, great. So who's suitable for a trial? Well, it really depends on the purpose of the trial. If it's an early stage study of a small molecule, it could be a healthy volunteer. If it's later stage studies, it would be patients. Are we looking at dosing, safety, or efficacy? And in terms of efficacy, there's different ways that it can shake out. Are we looking to prevent disease? In that case, we wouldn't want to give treatment before the disease symptoms are very severe. If, on the other hand, we're looking to show improvement in a disease, then patients should have symptoms in order that we can see a change. And then finally, there may be cases, especially with neurological conditions, where stabilization or slowing of disease progression is really the goal of treatment. So how do we know if you're eligible for a trial? Well, a good place to start would be a website called clinicaltrials.gov, which lists most of the ongoing clinical trials in the US. It'll tell you which trials are active, which ones are enrolling. It'll give some basic information about the disease, the drug, the inclusion criteria. And importantly, it'll have a contact number for the study site staff so that you can contact them. You can meet with the investigator, hear more about the study, discuss the risks, and if you're interested, then you would sign the informed consent and then undergo screening for eligibility. So who should participate in a clinical trial? Well, there's no right or wrong reason to either participate or not participate. It's really a personal decision for each family depending on their circumstances. Things to consider would be the disease severity and whether, the prognosis, would indicate a very rapid disease progression or a slower disease progression. Are there other treatments that someone could be taking before this particular drug is approved? What are the risks? Maybe they're too great, for someone to consider. Is this going to be a chronic treatment where there's an expectation that you would take a drug every day or once a week? And you could stop treatment if you wanted to, or is it a permanent treatment like gene therapy or transplantation where you can't necessarily stop the treatment, it'll be with you forever. And then also the family situation, participating in a study requires a commitment from the family, and there needs to be an adequate support system for the patient and typically a caregiver to travel to the site to participate in the different clinic visits. Now, the good news is that clinical trials are free, or they should be free. Patients are doing a service to help identify whether or not a drug is safe and effective. The drug is free, the investigational drug is free, the hospitalization, the assessments that are done during the trial are free, and typically any travel-related expenses for a patient and a companion are also covered by the clinical trial. Now, something that's different in a clinical trial is expanded access. Sometimes people call this compassionate use, and this is when a patient receives a drug outside of a clinical trial, and there are special criteria that need to be met to be eligible for expanded access. And basically, the patient has to have a life-threatening or serious medical condition with no access to either a satisfactory treatment or to a clinical trial. And that may be because there is no trial ongoing, there may be a trial but they're not eligible, or a patient may not have access to treatment. Now, an IND could be put together for an individual patient, but it does require cooperation of the physician, the company, IRB and the FDA. And I think the main concern with expanded access is that either there may be not enough drug to treat patients outside a clinical trial, or participating or patients who receive drug outside a clinical trial might interfere with the main trial actually being able to enroll patients. So expanded access, while it is available, it's not something that's done on a routine basis. And then the last question relates to COVID-19. It is slowing down enrollment in trials. It is disrupting trials. Sites are communicating with FDA about how to minimize the impact, including use of telemedicine or remote assessments or accessing medical records. FDA has provided a guidance on how to deal with the COVID-19 impact, and communication between the sponsor and the FDA is paramount to maintain patient safety during a trial where all the safety monitoring may not be able to take place, or to maintain integrity of the trial. And then finally, answer the question, how long does it take a drug to get approved? I'd say on average, we're not looking at months or years, but probably about 10 years. And the reason for that is that there's a lot of legwork before clinical trials take place in terms of drug discovery and optimization, testing in animals, developing a manufacturing process. And with all that information, the FDA reviews it, and if found to be reasonable and allow a phase 1/2 clinical trial to start. That's probably about 3 years in. After that trial finishes, there's additional discussions about the next trial. What would it take to get a drug registered? And that can take a couple of years. And then there's also the review at the end of that trial that can take 6 months to 12 months. So, 10 years plus or minus a few years on either side, depending on this particular circumstances, is what we're looking at to get a drug approved from the beginning to the end. So with that, I'd like to close. And if you have specific questions about specific trials, I would encourage each of you to contact the study site investigator to learn more information about a particular clinical trial. So thank you very much. Back to you, Jamie.
Thank you, Dr. Cox. Okay, so our next speaker is Dr. Simpoku from the FDA. So if I could ask you to unmute and share here you are. Hi there.
Hi, can you see me and hear me okay?
We can see you, we can hear you. And if you want to start sharing your slides.
Sure.
And again, Dr. Simpoku, thank you so very much for taking time to join us today. I know I'm eager to hear from you, as are all the families joining.
Sure, of course. So let's see, hold on. Okay, you can see my slides now?
Yep, all set.
Great. So first of all, thank you very much for the invitation. This is a real pleasure and privilege for me to be able to talk to the group. I am currently the acting deputy division director in our newly formed Division of Rare Diseases and Medical Genetics within the Center for Drugs at FDA. I'm going to give you a very high-level overview of what we do and how we do it at FDA in general, and with some specifics on some considerations in rare disease drug development as it applies to the diseases that you are involved in and study, and give you a little bit of highlights of how we approach drug development guidance and review for different products. This is my typical disclaimer that I have nothing to disclose, and these are my own opinions. So this is quite basic, but it is obviously very important. And what does the FDA do? And so, in general, really, it is the organization whose role is to protect the public health. And how do we do that is by ensuring that, drugs or biological products or medical devices that are approved are safe and effective for their intended use. Now, the organization of FDA is quite complex, to say the least. This is just a very kind of bird's eye view of the organization. You can see my pointer. This is where the commissioner sits, and then under that, there's multiple offices and centers. There are a total of 6 centers. Our center is the Center for Drug Evaluation Research right here. And Dr. Rachel Whitten, who will speak after me, is in Center of Biological Evaluation Research right here. So CDER, or Center for Drugs, is a center which reviews and regulates products that are small molecules, so drugs, but also some biological products such as enzyme replacement therapies. Now within CDER, there's again multiple different offices. The Office of New Drugs is where all the staff and review divisions reside, and those are the ones who really review applications for new drugs, unapproved drugs, and also follow approved drugs in terms of safety post-approval. Now, within our OND, which actually was recently completely reorganized in terms of structure, our division is right here, Division Rare Disease and Medical Genetics. We are actually a brand new division, and we came together recently to form, so one of our responsibilities, the review for products of inborn errors of metabolism, and also including the rare disease program, which is tasked primarily with policy and guidance development. So this is just a little bit about our division. So we are involved in really the review of applications for inborn errors of metabolism. We consult on rare disease applications across OND and other centers and offices. We're involved in developmental guidances, and those are documents which explain or describe the FDA's general thinking on different concepts in drug development, in an effort to help developers engage with patients and patient groups via different ways. And then we collaborate with other regulatory agencies, including European Medicines Agency, Health Canada, and others. And this is especially important in the rare diseases where we know there's limited number of patients, and most trials, if not all, are global trials. And so consistency and harmonization across different regulatory agencies becomes important, and we really try to do that on a regular basis. Now, in terms of the goal of drug development, again, very basic, but really important, is really to gain scientific evidence that a drug is safe and effective for a particular use or in a particular disease that was studied. And really, that's the best access really to safe and effective drugs when you have a drug approved on the market. The reason for that is that for a drug to be approved, there has to be specific scientific evidence to support that it is safe, in humans, of course, primarily, but also that it has an effect to really justify the risks associated with its use. So the drug development process, Jerry talked about this a little bit. It's quite complex and takes a long time, a lot of resources, but there's reasons for that. All of those pieces are really important, and each in its own way, to really put the puzzle together. So the non-clinical testing really involves animal testing, in vitro testing in the laboratory. One of the most important elements in non-clinical testing is to identify safety risks in animal studies. Those are really important and need to be completed, many of them, prior to really going into clinical trials in humans, so that we know really what to monitor for in terms of safety, especially as it relates to really serious safety risk or life-threatening complications. The chemistry manufacturing process is also complex, but it really has to do with how you produce a product, how you manufacture it, what kind of elements for assessing its potency and its purity, and basically making sure that what you're manufacturing is the actual drug, and it doesn't include any other additional molecules that may create safety issues, for example. And of course, the clinical development is really the trials in humans. So those are clinical trials that are in different phases. Jerry mentioned that a little bit. In order to, for developers and sponsors to actually do trials in humans, they have to have an investigational drug application approved by FDA. I will talk a little bit about what that entails in just a little bit. And this is required for any trial of investigational drugs in humans in this country. And then the last piece is really when everything comes together after a long time and a lot of testing and data collection, the sponsor developer may feel ready to actually submit a marketing application, really application that includes all this different data for FDA to review and make a determination on approval or not. General schematic of the drug development phases. So you have kind of this pre-IND phase right here. That's before the developer submits an IND to FDA. And then the actual IND phase, which is basically from the start of any trial in humans until the time of a marketing application submission. So NDA is a new drug application, BLA is a biologic license application. It's just a marketing application acronyms. Now, in the pre-IND phase, as I said, is all the non-clinical testing, discovery, manufacturing, chemistry. IND phases have to do with clinical trials. The objectives really is to initially assess the safety in healthy volunteers and then in patients. You're trying to identify any serious, life-threatening and non-serious adverse events. The reason for that is that you want to see you want to see what are those risks that you need to monitor for, how often, and what needs to be done for that. And then really the phase 3 is to assess the efficacy in the patient population. In terms of when a sponsor is ready to submit this initial IND application, there are certain elements that need to be submitted with that. That FDA and our division reviews in detail to really make a determination whether we think that this IND is safe to proceed, is how we call it. So we need to have all the non-clinical or animal testing information to really inform the safety of the drug. That comes from animals, and there's specific requirements for that. And the reasons for that is that you want to assess both immediate risks and later onset risks. And really focuses on any serious risks that would have the potential of becoming life-threatening. We need chemistry, manufacturing information, the actual drug, and then elements of the trial design depending on what phase of the trial it is. So how does the sponsor and the investigator plan to do the safety evaluation? How often do they monitor and for what events based on what we know already about the drug? And what is the actual design of the study? What doses they plan to use? How fast they plan to go up or down? How long is the trial? Is this patients or healthy volunteers? And then what are really the objectives and how do they plan to meet those objectives in terms of assessments? Now, the primary review and primary focus on IND review is safety, because really it's the first in human or sometimes it's the first in human dosing of drugs. So there's a lot of focus on safety. However, if it is a phase 2 or 3 trial, which is this late stage development, which sometimes we may see, we are focusing both on safety, but also on the efficacy assessment. A little bit about the general principles of how really we base our decisions on approving a drug or not. This is very, very detailed, in a detailed manner, described really in the laws and regulations. And so the way that effectiveness is established is really about what we call substantial evidence. So there has to be scientific evidence that a drug really has the, the effect that it's, that the sponsor claims to have based on data that are, and that data comes from these adequate and well-controlled investigations. So these are almost buzzwords, and I underline here because they are really, really important, and really, those are really the fundamental tenets of what we review in those applications and how we do our work. So this is a busy slide, but I just wanted to highlight that there's specific elements of adequate, well-controlled trials that we look for when assessing the trial to basically ensure that it meets its objectives and its goals, and that it's not just giving a drug to a patient population without really have any prospect to obtain informative data, because of course that wouldn't be ethical in general. So the first stage is really the ability to distinguish what is the effect of the drug versus what is the effect of other influences. So other influences may be actually a natural disease progression. If you know the disease natural history and you know that over a certain period of time, a certain aspect of the disease may get worse or may get better, that's really important information because you need to really make a determination of whatever change you saw. Is it a drug or is it something else? How we do that is by having a control group, right? So I think that was mentioned previously, a control group is really to make comparisons. The bottom line in many clinical trials, and when you really review data, is comparing it to what would have otherwise happened if patients did not get the drug. So that's why we believe that control groups are really, really important. Otherwise, we are not able to really make sense of whether what actually happened in a trial is due to the drug versus something else, versus the disease itself, versus the way they actually assessed the patients, and other, and other considerations. There are multiple different types of controls. One is a placebo control. Another is just no treatment, standard of care. There are historical or external controls of different ways to do that. But really the gold standard is to really use a placebo over some duration of time to be able to balance the groups, to be able to compare them. The rest has to do with what population you're selecting. Is that population going to be informative in getting you the data that you need? Appropriate assignment of patients. So you need to be randomizing, and that really has to do with balancing the groups in terms of what you think may actually affect the actual outcome. Now, how do we think about effectiveness? It really has to do with benefit, right? So really is the impact of treatment on how a patient feels, functions, or survives. That's really what it comes down to. And so that means that the drug, we have to have evidence that the drug has a positive impact on survival, so how long a patient lives, or how a patient feels in their daily life, or how the patient functions in their daily life. Those are just very clinically meaningful outcomes that we look for, and for good reason, obviously. So there's different ways to assess that. Obviously, assessing survival is the most straightforward, but this is not an endpoint that we'd like to use, obviously, in rare diseases, because we want to be able to get a treatment out there before having to get a survival benefit. So what we look for primarily is how patients feels or function, and that's assessed with clinical outcome assessments. So these are just tools to track symptoms, performance and tests, or clinician observations. Another way to define effectiveness in some cases is showing an impact on a biomarker. There are some or many inborn errors of metabolism which have associated biomarker changes with them. So GM1, GM2, and AA, and some others are ones that are really very disease specific. In some circumstances, those could potentially be used to establish effectiveness, but there has to be a very thorough review of the available information to really support that. I have to say that in many of our inborns of metabolism, we're not quite there yet to only rely on a biomarker or laboratory marker to provide evidence of this efficacy. So it's very important to really assess the feel and function in clinical trials. So now in terms of when a marketing application comes to us, what is kind of the general framework of our review? So there's different teams involved, many different ones. So the clinical team is composed of physicians. We have the clinical pharmacologist, the toxicologist who review the toxicology data from animals, the chemists who review really the chemistry of the drug and the manufacturing. So all of this is submitted in a huge data package. We review the actual raw data in detail. This takes a few months, between 6 and 12 months, depending on the application. And then in terms of the clinical review, it's really assessing the observed clinical benefits and really weighing those against the observed safety risks of the drug. Inspections are also involved in terms of the trial science to make sure that what they're supposed to be doing, they actually did. There is no, reliability of the data that are a problem, and then inspection of the facilities for manufacture. So my couple last slides, and I'm almost done, is really what are the essential considerations when we assess a drug or assess a marketing application to determine whether a drug should be approved or not. So first of all, the data that were collected, were they rigorously collected and they actually reliable for us to be able to make a determination? So that has to do with the design of the trial, the population, the actual tools they use to assess this, and how well did they assess the safety of the drug and actually put in measures to address some of the safety issues. Second is, are the observed clinical effects meaningful to patients? So that's a really, really important point that we most times stress to sponsors and developers. And of course that comes from patients, right, and patient groups, is to hear what do they think is meaningful to them. It might be that what is meaningful to change in one patient may be different than the other patient, or what is meaningful to me may be different than what the actual patient thinks, or the industry and the developer. So, you know, there's a very big effort at FDA for many years focusing on patient-focused drug development. So this basically has to do with gathering information on what really matters to patients, how to assess that in clinical trials, and how to really provide data to support that it's meaningful for a patient in terms of feel and function. Other is, are there available approved therapies, and is there unmet needs in the community? Is this the first drug that will go on the market versus the 15th or 16th drug, where the review may be a little bit different? And are the safety risks serious or life-threatening? So really that becomes important when assessing the safety of the drug. And are those safety risks able to be monitored and treated adequately or not? At the end of the day, the team has to make really a benefit risk determination. So once we review all the data on efficacy, and we have reliable data, and we think maybe something is clinically meaningful and the drug is doing what it's supposed to do, or the developer claims to do, we have to really weigh that against the safety risks that are identified. Is there a favorable benefit risk for the patient population or not? And that really becomes kind of the final determination of whether a drug is approved or not. There are many special programs that are specific to promote drug development in rare diseases. FDA offers financial incentives. One is the orphan drug designation, rare pediatric disease priority review vouchers. Those are all financial incentives to develop drugs in rare diseases. And the last piece is the expedited programs. Those basically provide frequent interactions and guidance from FDA review divisions for specific products that have proven to be very promising in specific rare diseases. The last two have to do with what we call accelerated approval. So that's an approval pathway for a drug using a biomarker and not this clinical outcome of the feel and function. There's a requirement, a legal requirement to do another trial to really confirm that benefit, in which we really need to have assurance with data that whatever the drug does is actually clinically meaningful in patients. The last one is priority review of a marketing application. We do see that often in rare disease applications, especially when there's first drug approved in the disease. The criteria for that are very specific. So if it is a drug that actually is providing you know, a superiority over other drugs that are potentially approved for the disease, or it's really one for a rare or serious and life-threatening disease, then we take much less time to review that to get it out of the market. So this is, I think, my last slide, and I'm wrapping up. The last piece about expanded access, I know Jerry touched upon this a little bit, and I'll be happy to answer questions. But really, just to emphasize, when it comes to expanded access, this is just a different regulatory pathway to obtain access to investigational drugs, which is only for treatment purposes. So typically, data that are collected through expanded access are not used in a marketing application. There are specific considerations and legal requirements for that. So these are really for patients who may not be eligible for enrollment in clinical trials. And then there's specific considerations for the safety and the prospect that the drug would have a benefit in patients. The last piece to emphasize is that these expanded access INDs or applications should really not interfere with development of the drug, which means that they really should not interfere with recruitment of patients into clinical trials, because that's really the only way that we can get informative data, right, to get products approved for the patient population. So this is something that we pay attention to a lot to make sure that the data that we're collecting from patients that are treated are really useful at the end of the day. And that's all I have. Any questions, you can get in touch with me in my email and I'll be happy to take questions.
Thank you so much, Dr. Simpuku. That was great. We do have a number of questions that have come in. We're going to save them for the end of our session. But again, as a reminder to those of you listening, please feel free to use the Q&A function to submit your questions. And now I'm pleased to introduce Dr. Witten, also from FDA, who's going to provide some additional information for us. Thanks, Dr. Witten.
Do you see my slides?
Yes,
Yeah.
we do.
Yes, hi, my name is Rachel Witten, and thank you so much for inviting me to participate in your annual conference. I'm gonna talk and discuss gene therapy for rare diseases. And you just heard Petrul's presentation about their CDER approach to rare diseases, to the clinical trials. CDER and CBER are very similar, and at the same time we're very different. And this is what, just let me see what happened with my slide. And yeah, we're very different, and I'm gonna talk about our differences. This is us, this is CBER, and the fourth person on the left on the picture is our center director, Dr. Peter Marks. The Center for Biology included only several centers, included three product offices. One is the Office of Tissue Advanced Therapy. This is what it's our office. Another Office of Blood and Research, and another office, Vaccine and Review. We evaluate many different products, but today I would like to talk specifically about gene therapy, in particular about the opportunity for more efficient drug development for gene therapies. The workload in our office is really, expanding, and compared to the 2016, we have like more than 100%. So our investigation new drug submission increased by more than 100%, and the same as research submission also increased by 95%. And the same with gene therapy. And gene therapy, if we have 67 submission in 2006, we have in 2009, we already have 161 submissions for rare disease and for gene therapy. And approximately our workload, it's in all the application, the investigation new drugs, it's for rare diseases, it's more than approximately 50% our workload. What is the gene therapy? Gene therapy is a technique that modifies a person's gene to treat or cure disease, then introducing foreign genomic material into host cells to elicit a therapeutic benefit. The key success of gene therapy is to create safe and efficient gene delivery vehicle. And this is when I show my slide what kind of gene therapies we have products. It's included many difficult vehicles, many difficult viral vectors. Gene therapy can work by several mechanisms: replacing a disease-causing gene with healthy copy of gene, inactivating gene causing gene that is not functioning, or introducing a new or modified gene in the body to help treat the disease. Let's talk about milestones and setbacks in gene therapy development. First gene therapy trial in 1990, a 4-year-old girl became the first gene therapy patient, and its trial was at NIH. She had adenosine deaminase deficiency, severe combined immunodeficiency, which left her immunosuppressed. The vehicle was used retroviral vector, and she received this ADA gene into autologous T cells. This girl she's alive and doing well. In 1999, I think it's a very well-known case, 18 years old Jessie had a rare metabolic disorder. It's called ornithine transcarbamylase deficiency syndrome, or OTCD. And this, when you have this disease, ammonia level builds up in the lethal and very high levels in the blood. And in this condition, the patient may, have this, may develop coma or just start vomiting. And Jesse was in a pretty good shape, and but when he became a teenager, he stopped taking this scavenger medication. And when the trial opened in Philadelphia, he was very eager to try. He was practically a healthy volunteer in this trial, and the vector was used to deliver. It's called adenovirus, and unfortunately, after he received the product, he died 4 days later. And the reason still nobody knows. It's possible that Jesse had been previously exposed to this adenovirus, and when he received this product, his body develops a separate reinfection, and instead of fighting this virus, also, they were thought that maybe at that time he had a very high ammonia level, but also when they started doing the investigation, the investigator did not disclose then in his preclinical study, 1 monkey died. And the news of experimental therapy and that killed basically healthy volunteer rocked the field of gene therapy, and the product would be and stop practically experiments with gene therapy for rare disorder. French researchers conducted gene therapy in 2002, and they used stem cell taken from the patient bone marrow. This patient was diagnosed with X-linked severe combined immunodeficiency, or BOI, and the Bubble syndrome, also a genetic disorder which the patient lacked most type of the immune cells. And this trial was very successful, and the kids were doing very well for 4 or 5 years. Unfortunately, 5 of the 20 treated children developed leukemia due of this intestinal mutagenesis, which is part of DNA, kind of went into another DNA, and it's T cells called leukemia. 4 of these patients were successfully treated with chemotherapy, but 1 patient died. And this is kind of its explanation why it took so many years for us to finally approve first 4 gene therapy product. In 2017, in August and October, 2 cell-based gene therapy for leukemia and for non-Hodgkin's lymphoma were approved. But in December 19, 2017, FDA finally approved Luxturna, the first gene therapy for an inherited disorder, retinal dystrophy, which is a rare condition that causes progressive form of blindness, usually beginning in childhood. In 2019, FDA also approved Zolgensma for type 1 spinal muscular atrophy in kids less than 2 years of age. This is already so like twice already. This is how long does it take to develop safe and effective product. Compared to CDER, we have in CBER, because our product is difficult and complex, we have, when a sponsor come to us with the idea of development of the product, we have the meeting, it's called Interact, when we're kind of discussing, it's not binding, but we're discussing and explaining to the sponsor what study he needs to do, what we're looking at the product. And it's most of this for the product and for this preclinical studies. And when you see yellow squares, this is how often we'll talk to our sponsor to try to be helpful in this drug development. Regulation of gene therapy, traditionally it was the domain of academic medicine. Recent increase also in large pharmaceutical companies, Park, Lysogen, and other companies. What we ask for, what we really would like to see in our regulation of gene therapy, we ask the sponsor to start right from the beginning. Get input from the rare disease patient network, obtain early advice from the different regulatory authorities. We also would like, and we usually tell the sponsor, then they should plan a global trial for reasonable enrollment rate, because the trial usually for rare diseases is very, amount of patients enrolled in this rare disease trials is very small and very difficult. Make determination of efficacy if you don't have enough patients, and of course collaboration. Why it's difficult to find treatment or cure rare diseases, especially we don't know what kind of endpoints we can use, what is the outcome should be measured, tools, instrument. Natural history often is not sufficient. We cannot get this data from the natural history. And we, because there's so much, every patient is so different, and it's very difficult to find clinical endpoints or surrogate endpoints, and they usually are not established. A database is efficacy and safety, but almost in a small trial will be small. This is what we would like the sponsor to enroll as many patients as possible, including global trials. And I mentioned, Petrula been just explaining to you what does this mean for phase 1, 2, 3. Like I mentioned, our trials a little bit different, our products a little bit different, and this is what I'm gonna provide some explanation. The product, manufacturing product, we have, the sponsor doesn't have too many facility, and they have to choose the right facility to make this product. And because this manufacturing, they have very limited experience, and it's very difficult to make many lots. It's in a short, and this product usually have a short shelf life, and which is very important to give a prolonged life, because most of the time this product has to be shipped to the place when it's going to be injected. What happens sometimes in our trial, the sponsor starts clinical trial, and then after phase 1, he figure out maybe I should do the different promoter or different product or use different route, and it's very difficult to even to get this data from, or at least do something with this data from the phase 1 trial. If it's a phase 2 trial, the sponsor wants to use something else. And this is, the comparability issue. It's a big deal for us. Dose exploration is limited by cost and manufacturing capability. To make this gene therapy product is outrageously expensive. And occasionally it's very difficult to make the concentrated product. It's mean if you want to give the higher dose, you have to concentrate the product, and it's very challenging. Pre-existing antibody to gene therapy product may limit its therapeutic potential. This is what happened with Jesse. He probably had this antibody and he did not react to gene therapy. And now for most of I will say for all of our trials, we're just checking for if it's supposed to use a specific vector, we're checking for antibody to this vector, not to repeat the mistake what happened in the past. And also gene therapy product may require invasive methods. And usually we need to develop this device or from the beginning, and you need to get the expertise from the different center, which is called CDER-H. It's was the evaluated devices. Sorry. Clinical pharmacology toxicology is, because this is not the drug, this is biologic product, and we do not allow to enroll healthy volunteers in our trial. We're trying to get this proof of concept from the animal model, because most of the time pediatric patients enroll in this clinical trial. And animals should be comparable to human and by pathophysiology, clinical sign, and responsiveness to the intended program. And we need to understand and discuss the limitation to the target patient population. Animal model comparable should be comparable physiological progression of the disease phenotype, disease etiology, disease pathophysiology, stage and severity of the disease should be matched to patient population. And clinical and traditional phase 1, 2, and 3 are commonly blended into early phase versus later phase. And we see more very common the patients, the sponsor originally open phase 1 and for evaluation of safety and preliminary efficacy, because you cannot give gene therapy just and without expecting to see some efficacy. And then collecting this data, the next step for most of our sponsor is the late phases for the clinical development. It's very critical to randomize patient, even the first in human trial. But we know that patients are reluctant to be randomized to standard of care that may be known to be not effective. And also being randomized to control arm, they may become ineligible for other studies to receive another gene therapy. And what is the what is the life of gene therapy? And we really don't know. And therefore, when natural history is well defined and expected treatment effect large, we may consider using a historical control as the control arm. But again, patient this natural history should be comparable to the clinical trial. Natural history should include important endpoints of this trial. Definition of variable in the same time in natural history and trial, and starting for potentially therapeutic dose. We just finished our guidance gene and cell gene therapy for rare diseases. We specifically mentioned that you have to use therapeutic dose in the clinical trial, not subtherapeutic, how it was decided long time ago. Now we want to see some efficacy from the beginning.
Dr. Witten, excuse me, it's Jamie. I just want to give you a 5-minute warning.
Okay.
Thank you.
And subject was rare disease. You cannot try multiple gene therapy. And again, for gene therapy, it's only one shot, at least at this moment. Okay, what is safety assessment? And we're very careful with evaluation of this clinical trial, because possibility of safety concern, viral shedding, immunological reaction to the vector, level of viral replication persistent on non-target tissue. And also we have the same concern of new gene editing technique. Maybe it's just because of off-target effect. What we're looking for approval, as Petrola mentioned before me, it's effectiveness and safety through the adequate and well-controlled clinical trials. Not necessary to be randomized, we would like to be randomized, but it could be just very well controlled natural history study, or it's armed with a standard of care treatment. In summary, genetic basis of human disease is complex, and we still have a lot to learn. Gene therapy products hold great promises as a therapeutic agent to treat for a treatment of wide variety of diseases. With the approval of the initial few gene therapy products, the high expectation for development of future application of gene therapy. FDA is committed to bringing the promise of innovative, safe, and effective new therapies to those in need of them as quickly as possible. But vigilance is essential, and we don't want to forget the lesson learned from the past and the clinical experience with gene therapy. The FDA has lessons learned, the industry has learned lessons, and we're all seeking to be very careful in how we advance. The risk is never goes away. That's what it takes to make a new medication, new medicine. This is our guidance we provided to our sponsor to help them to design clinical trials for gene therapy. And this is us. If you have any questions, please email us or give us a call. Thank you.
Excellent. Thank you so much for that very helpful overview specific to the gene therapy division.
I don't know how to close it. If you
Okay.
can,
I think the producer can help us close the slides. So we did have a number of excellent questions, and I want us to try to get to them. So while we're navigating, just resuming the video for the three panelists, I may just answer one question that came in around registries and the NTSAD registry. So just an update from Sue Khan. So there's no single NTSAD disease registry. There is the NTSAD database where we keep basic contact and disease information for members. NTSAD participates in the Patient Insights Network for each of the diseases through Invitae. And NTSAD has also participated in a number of retrospective and prospective natural history studies, which have led to publications that have been very helpful and informative. And going forward, as you saw at the beginning in the research priorities, figuring out how we can integrate all of this information together is one of the activities that group is hoping to support moving forward. So I hope that answers the question about NTSAD registries. I know there was another question asked about natural history and in the context of gene therapy. And I think if I'm understanding the question correctly, natural history is not specific to any one particular therapy. It's really a look at the disease over time without a therapeutic intervention, be it gene therapy or something else. So now when we talk about natural history, we're really wanting to see what is the natural course of a disease, and that information is critically important, frankly, in rare diseases to help sponsors and other researchers as they develop different protocols and look to show potential benefit of therapies being developed. So I'm going to start with a couple of questions came in around use of placebo. And I think, specifically related to the fact that, when we're talking about severe, very rare diseases with a small population that are very severe and progressive, the thought behind why one would have a placebo. And I know it was talked about a bit, both by Dr. Simpoku and Dr. Cox, but if maybe you want to just, Dr. Simpoku, maybe you can just, again, kind of quickly summarize why a placebo sometimes is necessary to assess a therapeutic intervention.
Sure, and this is an important question actually, and one that is really important to understand. So, really the reason for doing the trials is to be able to get information on what the drug does. Does it work or not? Is it safe? So if you only have patients who are treated and nothing else to compare really any outcomes, it becomes exceedingly difficult to really know, is it the drug that's doing what we are observing, or is it something else? So that really is it really is a fundamental question that we're trying to answer here. And the reason that we do trials and we do, all the statistical testing that's associated with that is to answer that question. So the reason why a placebo arm, so to speak, in a trial is important is because you want to have something to compare to. So a placebo is simply, an arm of, hopefully same or similar patients who are not treated to really be able to compare, is it the drug or is it something else that's doing what we're observing? Now, we do recognize that there is an urgency in these diseases, and I have certainly seen also myself in practice that those diseases are really devastating for the patients, for the families. Now, we, as an organization, as a division, and really based on our legal authorities, we are able to exercise what we call flexibility, right? So there is, in the law and in our regulations, it's really recognized that some diseases are very rare and some diseases are really serious to the point where we don't have really the luxury of going through the typical drug development and really being able to recruit many patients and have all the time that we want for drugs. And, Tay-Sachs and other associated diseases are a great example of that. So I would say that we recognize really the urgency of getting treatments out there, especially in those life-threatening diseases. There's certainly a lot of flexibility that we exert in terms of what kind of data we would accept, what kind of trials may really support this. We're not kind of have this blanket approach of if it's not a randomized placebo trial, we don't accept it. I don't think that's true. I think it really has to do with the drug itself, the population that they're trying to treat, and what exact symptom they're trying to treat. So, and that's why we really engage in a lot of discussion and guidance to developers and companies to really understand, and come up with some creative ways to do that. I don't know if that answers the question.
Thank you. Very, very helpful. Dr. Cox, I don't know if you wanted to make any additional comment on that, but I also wanted to ask you a question. If you could just maybe sort of remind everyone, when it comes to clinical trial participant selection, can you just sort of give a snapshot of how that process occurs, who ultimately decides who may or may not be entered into a clinical study?
Sure. So just, I think addressing the placebo question, it's a well-known fact that patients who believe that they're receiving a treatment that may help them will actually experience an effect where they feel better or they may perform better because they believe that they're receiving some type of benefit. And is really, I think, a surprise to everyone when initial studies were done where people were given essentially a sugar pill, and yet there was still a response that was seen, and that's been replicated over and over and over again. So in particular, when we talk about clinical benefit, and the basis for approval being, feeling better or, performing some kind of activity better. If you feel better about yourself because you believe you're getting treatment, there will be a certain amount of potential improvement that is just attributed to your willingness to believe that you're getting better. And so it's really important to tease that out, from what the true effect is of a drug above and beyond that placebo effect. And in terms of eligibility criteria, it really, comes down to what is the most appropriate patient population that will achieve the objective of the study. And so in some cases, the study is to determine the appropriate dosing, in which case you may need patients from different age groups, or maybe an older age group would have to be enrolled first. Perhaps there's an assessment like a 6-minute walk test where a patient has to be able to walk. And so either younger patients or those in wheelchairs might not be eligible if that's the main criteria for evaluation. So, it really comes down to what the overall objective of the study is that determines, what are the characteristics that may allow a treatment effect to be quantified. And then conversely, there may be some other confounding factors about a patient that might interfere with the interpretation of the study. So for example, if someone is on another experimental medicine at the same time, it would be hard to tease out what change is occurring due to which medications. So often there always is an exclusion for other experimental medicines being used up until the time of the clinical trial. There might be some other underlying disease states, like I mentioned, liver disease or kidney disease or cancer that might interfere with patients being able to fulfill the requirements of the study.
Thanks, Dr. Cox. Dr. Whitten, a question for you. Is it possible to make an accelerated pathway request before phase 3? It seems a little bit late in the process, was the comment. So do you have any insight on the opportunity for accelerated pathway or some of these expedited pathways?
We do, but when the clinical trial starts, we, like I said, we're helping our sponsor to go through, to help him all the way. But accelerated approval, it's mean expanded access, or just, I'm not sure I understood, because for accelerated approval, we see efficacy. We also have a program, it's called regenerative medicine advanced therapy. When we see the efficacy, preliminary efficacy in a clinical trial, we granted, it's called regenerative medicine advanced therapy, RMAT designation. And by getting this designation or breakthrough designation, eventually at the end, this product can go for accelerated approval.
Okay, thank you. And I know in the slides, there were a couple, there was an overview of different techniques that FDA now employs to help, expedite treatment. So people can go back and review those. And just a sort of a technical reminder that all of these sessions are being recorded. And so you'll have ongoing access to watch this again or focus and hone in on areas that were of most interest for you, just so the audience knows. Okay, another question around for the FDA, and I'm not sure which of you might want to try to tackle this, but an acknowledgement that manufacturing for gene therapy is very expensive. It's a very involved, complex process. The difference in cost between non-GMP manufacturing and GMP manufacturing is significant. What is the FDA's position for possibly allowing non-GMP manufacturing for at least a phase 1 study? And maybe you can also just clarify for the audience what non-GMP and GMP means.
I'm not a product reviewer. I'm not sure, I don't know how to clarify this. But because of the gene therapy is booming, it's not enough facility to make this product. And some sponsors have been waiting for 6 months to get to manufacture the product. And this is why it's kind of takes a long time or some even sponsors decided to build their own manufacturing product. And this is what's happening in this gene therapy field. We have this efficacy, we have the product, we have good results, we have efficacy, we have good results, but we don't have a product to approve.
Okay. Dr. Simpoku, anything else that you wanted to add, or Dr. Cox? Or manufacturing?
So I guess one comment on the accelerated approval, because it really becomes very confusing for people and even for us who actually do this job a lot. There's all the different terminology. So accelerated approval, really all it means is just what is the basis for approving a drug? Is it based on a clinical outcome, meaning how a patient feels, how a patient function, or is it based on a laboratory assessment or an imaging, really something that does not have to do with a clinical outcome. So all it is, it's really not accelerated, meaning that it really accelerates the approval of a drug, although it does get it in the market maybe earlier. But really it gets it out in the market almost on a conditional basis, right? So it gets it out faster, but there's still a requirement that the sponsors need to conduct a trial, right? To gather the data that are needed to prove that some effect is really meaningful. So in terms of requesting accelerated approval, I think that the discussions about accelerated approval can happen anytime during project development, before phase 3, during phase 3. Typically, we want to make sure that we have some agreement before they start, an efficacy trial, but really it's a discussion throughout the drug development. And so it really has to do with kind of really having evidence that whatever the marker or biomarker may be, is truly something that there's a high potential that actually will reflect something that's clinically meaningful to patients.
Thank you for that. I know we're running short on time. There is one other, and there's a number of questions I fear we're not going to get to. So again, the reminder that if your question was not answered either live or in writing, so maybe go back and chat because Dr. Cox went through and did answer a few of these, feel free to follow up with the NTSAD team. But there are a number of questions focused on expanded access, obviously because of the severity of the diseases that we're talking about here. And so while currently there are no expanded access programs available, for these diseases, I'm not going to focus on some of the specific questions that were asked about who might qualify, et cetera. But there is a question for FDA here on, is there any incentive that the FDA provides to sponsors when looking at treatments for ultra-orphan diseases to include an expanded access arm? Is that something that FDA sort of weighs in on or provides feedback on, or is that decision left to the sponsor?
Yeah, I can start. This is Petrula. So this is not a decision that FDA makes. The sponsor, I guess, has to make a decision of whether they would want to provide their drug for expanded access, meaning just for treatment purposes. They would need to discuss that with the FDA to get agreement on that. But it is their decision. We cannot make them do that if that's the question. But really, from our perspective, expanded access is only reserved in extenuating circumstances. It's really not the traditional way to develop a drug. And actually, if there's too many requests for extended access, especially in a very rare disease, it can actually really hinder the development of the drug. And so that's something that, sponsors really need to be really careful about. And also patient groups need to be very cognizant about. So for example, if there's patients who have no way of getting to the clinical trial site because of geographic considerations, because of whatever that may be, those patients may be considered to just get the drug for treatment purposes, or it might be that they're not truly eligible for a trial. However, what we really stress to the drug developers that come to talk to us is if there's any way to include a patient in a trial, they should do that. Is it through loosening the eligibility criteria, making it more broad to be more inclusive, and really stressing the fact that expanded access will be the last resort. And the reason for that is we want to make sure that we make maximal use of all the patients that we have in rare diseases. So the best way to do that is really include them in the clinical trial protocols, because that's really the best way that we can actually get informative data to approve a drug or not. So expanded access would not be a way to get a drug approved, if that's the question.
Mm-hmm. And I saw one question about the enrollment in gene therapy trials, what patient population, and regarding GM1 and GM2, somebody asked, we do not force, and the question was, why do you enroll younger patients? It's a sponsor choice who do enroll in this clinical trials. And because we would like for sponsor to enroll stable patients, not necessary to be youngest or oldest, but they have to be on stable condition because we're still looking for the side effect from the gene therapy. And we cannot tell sponsor you have to evaluate GM1 or GM2 if the sponsor provided good preclinical data, the product is working for GM1 or GM2. This is what the sponsor proposed to treat or at least to enroll in this clinical trial. It's not our decision, but it's our recommendation to enroll stable patient just to, especially in the first phase 1 study, to evaluate for the safety of this gene therapy.
Thank you for that. Okay, so I know we want to be able to give everyone a break between the close of this and the research breakout sessions. So I would like to just very much on behalf of NTSAD thank our 3 presenters and panelists. This was extremely informative, and especially for those of you, Dr. Witten and Dr. Simpoku from the FDA. We know how busy you are at the agency, and so to take your time to help educate us and participate just really means a lot. And certainly, we know how to reach you, and we know the FDA has a lot of opportunity for patient voices to be heard. So, thank you for representing FDA's commitment here at this conference. If we can put up the final slide, just so people have a view into what's coming up, what's next. So the breakout sessions start at 2 p.m. Again, you can access the links to those from the morning email or through the conference app. If you were not able to have your answer for this panel addressed, please feel free to follow up with the NTSAD team. And I wish everyone a pleasant afternoon, an informative afternoon. I think it's wonderful to have all the different organizations that are involved in research and in potential therapies here with us today. So ask your questions there as well, and stay well, and we will talk to you all soon. Thanks very much.
Thank you.
Thank you. Bye-bye.
Thank you so much.
Bye.
Bye.
Thank you, Jamie, and to all of our panelists. Thank you to all of you, our participants. I will be closing out the session in just a moment to get prepared for our breakout sessions. Have a great rest of your day, everyone. Bye-bye.